All right, get ready, because today we're taking a deep dive into some seriously cool science. You sent over this article about extending telomeres, and let me tell you, this stuff is fascinating. We're talking about those protective caps on the ends of our chromosomes, and I think I've got a pretty good analogy to help visualize them.
Oh, I'm intrigued. Lay it on me.
Okay, imagine those little plastic tips on your shoelaces, the ones that keep them from fraying. Those are called aglets, and they basically serve the same purpose as telomeres. They protect the structure from damage and degradation.
That's a perfect analogy. Those aglets keep everything nice and tidy, just like telomeres help keep our chromosomes intact and functioning properly.
Exactly. But here's the thing, just like shoelaces eventually wear down over time, so do our telomeres. They naturally shorten as we age, and that shortening plays a big role in cell senescence.
Yes, cell senescence. It's basically what happens when cells get old and tired, losing their ability to divide like they used to. It's a key process linked to a whole bunch of age-related diseases and even plays a role in conditions like Duchenne muscular dystrophy, where cells wear out much faster than they should.
Right, so finding ways to safely extend those telomeres has huge implications, not just for research into aging, but potentially for treating some of these diseases as well.
Hmm.
But that's where things get a bit tricky, isn't it? I mean, current methods for telomere extension have some pretty significant drawbacks.
That's right. Many of the current methods rely on using viruses to deliver the telomerase enzyme, the one responsible for lengthening those telomeres. But viruses can be a bit unpredictable. They can integrate their own genetic material into the host cell's DNA, and that can have unintended consequences. We could end up with cells that become immortal, and that's not exactly what we're aiming for.
Yeah, that's definitely a scenario we want to avoid. But this new research we're diving into today seems to have found a way around that problem. They're using something called modified mRNA to deliver that telomerase enzyme, TERT, into human cells. And this is what makes the research so groundbreaking.
Absolutely. Modified mRNA is a game changer. Think of it as a temporary messenger. It delivers those crucial instructions for making TERT, but it doesn't mess with the cell's DNA permanently. It just delivers the message and then gracefully degrades, leading to a temporary boost in telomerase activity.
Okay, so it's like a hit-and-run approach, delivering those instructions and then getting out of there before it can cause any lasting changes.
Yeah.
Pretty clever. And they tested this on fibroblasts and myoblasts, cells that are known to have a limited capacity for replication. They can only divide a certain number of times before they reach that senescence stage we were talking about. So what happened? Did this modified mRNA actually do its job?
Well, the results are pretty impressive. They found that the treatment significantly increased TERT protein levels in both types of cells. And importantly, it led to a measurable increase in telomerase activity. But here's the key thing. It was transient, just like we were hoping. That activity peaked at 24 hours and then went back to normal levels by 48 hours.
So a temporary boost. But did that temporary boost actually translate into longer telomeres? Did those protective caps actually get extended?
They did. They observed significant telomere extension in both the fibroblasts and the myoblasts. And what's really remarkable is that the extension was comparable to what you might see with those riskier viral methods, but crucially, there's no permanent DNA modification. It's like hitting the pause button on telomere shortening, at least for a little while.
Yeah, that's still pretty amazing, even if it's temporary. Being able to safely extend those telomeres opens up so many possibilities. It's like giving those cells a little bit of extra time to function before they hit that wall of senescence.
Exactly. And there's another detail from the research that I think you'll find fascinating. They used a technique called QFISH, which allows them to visualize individual telomeres within a cell. What they found was that after the treatment, there was a shift in the frequency distribution of telomere lengths.
Okay, that sounds a bit technical. Can you break that down for me? What does that actually mean?
Sure, essentially they created a distinct population of cells that had longer telomeres compared to the untreated cells. So the treatment wasn't just extending every single telomere uniformly, it seemed to be having a more pronounced effect on a certain subset of them.
Interesting. So some cells were getting a bigger boost than others, like they won some kind of genetic lottery and got a longer lease on life.
That's a great way to put it. And this raises some interesting questions about the dynamics of how telomerase interacts with individual chromosomes and what that means for how cells behave. But let's move on to how this treatment actually impacted cell senescence. I think you'll find this particularly exciting.
All right, let's dive into that. Did this treatment actually manage to delay the aging process in these cells?
Well, they looked at the classic markers of cell senescence, the telltale signs that a cell is aging, And what they found was that in the cells that had been treated, there was a delayed onset of these markers. For example, they looked at an enzyme called beta-galactosidase, which tends to be more active in aging cells. And what they found was that it took longer for the treated cells to start showing high levels of this enzyme.
So even though the telomere extension was only temporary, it still had a lasting impact. It pushed back that state of senescence.
Precisely. It's like giving those cells a bit more time to function at their best before they start to decline. And they also looked at cell size, another marker of senescence. Aging cells tend to get bigger, and they found that the treated cells stayed smaller for a longer period of time, again suggesting that aging was being delayed.
Okay, so they delayed the aging clock, but did they stop it completely? Did these cells become immortal?
That's the million-dollar question, isn't it? And the answer, thankfully, is no. They didn't escape that fundamental limit of a cell's lifespan. The treated cells did eventually stop dividing, just like the untreated ones. And they did eventually start expressing those senescence markers, just like the untreated cells.
So it's not about creating immortality, but rather extending that healthy, functional period of a cell's life. And when you think about potential therapeutic applications, that's probably a good thing.
Absolutely. The goal is to enhance cell function, not create something completely unnatural or potentially dangerous. And this controlled, temporary nature of the telomere extension seems to strike a really good balance. But I think it's time we dig a little deeper into the techniques they used in this study. I think you'll find their approach quite clever.
Okay, I'm ready for the next layer of this deep dive. Let's get into it.
Welcome back. Now, remember how we talked about modified mRNA being used to deliver instructions for making more TERT? Well, the researchers had to actually prove that was happening. They had to show that those instructions were getting through and increasing TERT levels inside the cells.
Right, makes sense. But how do you even measure something like that? It seems pretty complex.
You're right, it's not exactly something you can just see with the naked eye. So, they used a couple of different techniques to confirm their findings. One of them is called Western blotting. Imagine it's like taking a snapshot of all the proteins present in the cells. This technique allows them to specifically visualize the amount of TERT protein that's there.
So they took a before picture and an after picture to see if there was more TERT after the treatment.
Exactly, and that's precisely what they found. Treating those cells with the modified TERT mRNA led to a significant increase in TERT protein levels compared to the untreated cells. To really nail down the results, they even used a control group treated with a catalytically inactive version of TERT mRNA. And that group showed no increase, confirming that it was the modified mRNA that was doing the work.
That's a really elegant way to prove their point. If you're delivering instructions to make more TERT, you should see more of it. Makes sense. But showing an increase in the protein is only part of the story.
Right, right?
They also had to prove that this extra TERT was actually forming a functional telomerase enzyme and doing its job extending those telomeres.
You got it. And to measure that activity, they used a clever assay called TRAP, which stands for Telomeric Repeat Amplification Protocol. It's designed to detect and measure the activity of telomerase in a sample.
Okay, so instead of just looking at the amount of TERT, they're now checking if it's actually doing what it's supposed to be doing, adding those repetitive DNA sequences to the ends of the telomeres.
Precisely. And the results were very clear. They saw a peak in telomerase activity about 24 hours after treatment with that modified TERT mRNA. And then, just like we saw with the protein levels, it returned to baseline by 48 hours.
Again, highlighting that temporary effect. A burst of activity, a bit of telomere extension, and then it fades away. No permanent changes, no risk of cells going haywire.
Right. But remember, just seeing more telomerase activity isn't enough. That activity has to actually translate into real changes in telomere length. So they had to find a way to measure that directly.
And how did they do that? Did they have some kind of tiny ruler for measuring these microscopic telomeres?
Not quite a tiny ruler, but they did have a very precise way to measure those telomeres. They used a technique called quantitative PCR, or qPCR for short. Think of it like a molecular copy machine that lets you measure the amount of specific DNA sequences in a sample.
So they used this qPCR to specifically measure the amount of telomeric DNA, and by comparing the amount in treated cells to the untreated cells, they could determine how much the telomeres had actually grown.
Exactly. And once again, the findings were quite remarkable. They observed a significant increase in telomere length in both cell types after treatment with the modified TERT mRNA. I know we've said it a few times, but it's worth repeating. The extension they saw was comparable to what you might see with those viral methods, but without the permanent modifications to the cell's DNA.
Wow, that's impressive. So they increased TERT protein, They boosted telomerase activity, and they actually made those telomeres longer, all in a way that's temporary and controlled. They've really ticked all the boxes.
They have. They use multiple techniques, and the results all point to the same conclusion. But I know what you're thinking. How much did those telomeres actually grow? Was it just a tiny, insignificant change or something more substantial?
You read my mind. I want the numbers.
They found that, on average, telomeres in the fibroblasts were extended by roughly 0.9 kilobases. And in the myoblasts, they saw an extension of about zero point 0.7 kilobases.
Okay, kilobases. I'm going to need some help understanding what that actually means.
Sure, to put those numbers into perspective, consider this. The average rate of telomere shortening in human cells is estimated to be somewhere between 50 to 100 base pairs per year. So an extension of zero point 0.7 to zero point 0.9 kilobases. That's basically adding back several years' worth of telomere length.
Wow, that's pretty significant, even if it's temporary. Being able to reverse that much telomere shortening in such a short time, that's a big deal.
It really is. It highlights the potential of this technique to rejuvenate cells. But there's another crucial aspect we need to address, and that's senescence. Remember, the whole point wasn't to make these cells immortal, it was to understand how this treatment affects the aging process. So how did they determine that these cells weren't becoming immortal or cancerous?
Yeah, that's a good point. They already showed that the treatment didn't make the cells divide forever. Did they do anything else to confirm that these cells weren't going rogue?
Yes, they did. They looked for very specific signs of cellular senescence, those telltale signs of aging. They used a couple of different markers, and one of them was an enzyme called beta-galactosidase, which tends to be more active in cells that are undergoing senescence.
So if those treated cells were truly immortal, we wouldn't expect to see those senescence markers showing up.
Right, and that's exactly what they observed. The treated cells, while they did divide for a bit longer, eventually started expressing those senescence markers, just like the untreated cells did.
So they delayed aging, but they didn't completely stop it, which again is a good thing if you're thinking about potential therapeutic applications. We want to help cells function better, not turn them into something completely different.
Exactly. The controlled, temporary nature of this telomere extension is a key factor in maintaining that balance. But let's switch gears a bit now and talk about what all of this means in a bigger context.
Okay, I'm ready to zoom out and look at the bigger picture. Okay, so we've seen that this treatment can temporarily extend telomeres and even delay senescence. But are there any other measurable effects on the cells themselves?
Yes, definitely. They also took a close look at cell size, which is another important indicator of senescence. As cells age, they tend to get bigger, and in this study, they found that the cells treated with the modified mRNA actually stayed smaller for longer. Once again, this points to a delay in the aging process.
So we're talking about multiple signs of a slowdown in cellular aging. Longer telomeres, those delayed senescence markers, and even a difference in cell size. It seems like this treatment is hitting aging from multiple angles.
It really is, and this research is not only incredibly fascinating from a purely scientific perspective, but it also has the potential to completely revolutionize how we think about aging and the possibilities for intervention. But I think it's time we step back from all those technical details and consider the broader implications. What does this research tell us about aging in general, and what might the future hold for this kind of technology?
Okay, let's shift gears a bit and talk about that bigger picture. I'm ready.
Well, I think one of the most exciting things about this research is that it challenges a very long-held assumption. For many years, we've viewed aging as this passive, inevitable decline, something that just happens to us, and there's not much we can do about it. But this study really challenges that notion. It shows that aging, at least at the cellular level, is a much more dynamic process. It's not just a one-way street.
So you're saying that aging isn't just a matter of fade. We might actually have some degree of control over how fast or slow our cells age.
Exactly. That's what makes this research so groundbreaking. We've known for a while now that telomere shortening plays a key role in aging, but this study goes a step further. It demonstrates that it's not just this fixed, irreversible process. We can intervene, we can extend telomeres, even if it's just temporarily. And that opens up a whole new world of possibilities for how we might treat those age-related diseases and maybe even extend lifespan.
It's like something out of science fiction, isn't it? The idea that we might actually be able to control or at least influence the aging process.
I know, it's pretty mind-blowing. But of course, with any groundbreaking research like this, there are still a lot of unanswered questions. There's much more to explore. And one crucial area for future research is looking at how this technique works on other types of cells. especially stem
The
cells.
stem cells are fascinating, aren't they?
Yeah.
They have this incredible ability to differentiate into so many different types of cells in the body.
Yeah.
So if we could extend the telomeres of stem cells, wouldn't that have huge implications for regenerative medicine? I mean, the possibilities seem endless.
Absolutely. Imagine being able to grow new tissues and organs for transplantation, or repairing damaged tissues in a way that's far more effective than what we can do currently. If we can figure out how to safely and effectively extend the telomeres of stem cells, it could revolutionize the entire field of regenerative medicine. But of course, we're not quite there yet. There's still work to be done. And another area of focus is going to be optimizing the delivery and the effectiveness of this modified mRNA treatment.
So making the treatment even more precise, more targeted, and maybe figuring out ways to make the effects last even longer.
Exactly. Right now, we're still in the early stages of this research, but the progress that's been made is incredibly exciting. It feels like we're standing on the edge of a new era, a new way of understanding aging and the possibilities for intervention.
It's really incredible to think about. And it's a perfect example of why we do what we do here on the Deep Dive, taking these complex scientific discoveries and unpacking them for you, our listeners.
It's a privilege to be a part of this journey of exploration, and we're always thrilled to have you along for the ride.
Thanks for joining us on this deep dive into the world of telomere research. It's cutting-edge science, and it's a field that's just bursting with potential. We can't wait to see what the future holds. Until next time, stay curious, keep asking questions, and never stop exploring. This is the Deep Dive signing off.